Well, good evening, everybody. Welcome. I'm Sterling Johnson. I'm the principal investigator of the RAP study, and I want to start by just thanking you for being here and thanks for being in this study. We're looking forward to telling you about the latest findings in the RAP study and also a little bit about what's happening in the field in general. And so this will all be happening tonight. You received a red bag tonight. It looks like many of you have starting to go through that. There's in that folder that you have in there is a copy of all of our slides. There's also a list of the publications that we have over the past several years. There's a blue piece of paper in there that has our contact information on it and a listing of some of our studies that we're going to tell you about tonight. There's also a green piece of paper that is an evaluation form so that you can tell us what worked tonight and what you'd like to see next time. Well, this study's been going on since 2001, and we have 1,550 or 1,560 people in this study now, and it's been yielding some incredible results, and we're looking forward to telling you about that tonight. We're going to start with an overview of the state of the field, and that'll be by Dr. Nate Chin. And following that, we will give you some of the RAP findings themselves, some of the new things that we've been discovering thanks to your participation. And then I'll come in and sum up and tell you about the next few years with the RAP study and what we'd like to do. Well, let me turn it over to Dr. Nate Chin, who's a geriatrician with our program. Dr. Chin. Careful of it. Careful.
Can people hear me? Okay. Thank you, Sterling. Well, good evening, everyone. I'm, as Sterling said, Nathaniel Chin. I'm one of our UW geriatric memory physicians. I'm also the medical director of the Alzheimer's Disease Research Center and the RAP study. And I'm really happy to be here, and I'm happy to see so many people show up today. The parking lot is full, which is always a good sign. And I'm going to go off script for just a few minutes and really start with a message that's on my last slide, which is thank you. And I'm not thanking you as someone with the titles that I just mentioned, but as the son of someone who recently died of Alzheimer's disease. I think about Alzheimer's every day, and its impact on me has been immense. I wouldn't be standing here today if not for my father and my mother. In fact, I may not have been a geriatrician at all. I had a different career path in mind, but like you all, I wanted to do something about Alzheimer's. I wanted to act. And so thank you all for acting. Now, I can't volunteer in the RAP study or any of the ADRC studies due to age. However, in all honesty, I don't know if I have the strength to endure what it is that you all do at every visit. And I appreciate the laughing, but I don't say that lightly. What you all do is courageous, and it's needed. We need volunteers like each one of you if we're going to prevent, treat, and cure this disease. And I say it in that order specifically. While I know that one day we will cure the disease, as someone directly affected by Alzheimer's, I'm still hoping for treatment and I'm still hoping for prevention and meaningful treatment. The ADRC and the RAP study are also focused on this. We share that goal, which is why I'm here, and I love working with all of you, though often it's really behind the scenes. I need you to keep doing what you're doing, and I thank you as someone with a family history. Now, with that said, the physician and researcher in me is here to ask for more, and I'm sorry I have to do that, but I need more from you if you can give it. I have the next 15 minutes to explain why, and it starts with understanding the disease and why we need biomarkers to identify brain changes in all of us. So the title of my talk is The State of the Field in Alzheimer's Disease, and I'll get right to it. So in the state of Wisconsin, there are over 110,000 people thought to be living with dementia due to Alzheimer's disease. And that number is expected to increase to 130,000 by 2025. And mind you, that number doesn't include the partners, the caregivers, the children, the friends, the neighbors. I truly believe this is a disease that's going to touch every single person one way or another. Now in 2015, over 2,000 people died from Alzheimer's disease in our state, making it the 6th leading cause of death. Not only does Alzheimer's disease have a large impact on the quality of life and the health of persons with dementia and their caregivers, but it has a huge impact on the healthcare costs as well. In 2017, Medicare spent over $19,000 per capita caring for people living with dementia. We spend about 1 in 5 Medicare dollars on this, and it's projected that by 2050 we'll spend 1 in 3 Medicare dollars on this. And those numbers are significant because it will bankrupt our healthcare system if we don't do something about it. So these statistics are just a few reminders of the devastating impact this disease has, not to mention the countless stories each one of us could tell and how this disease has affected our families. And that's why we're here today, to partner together to find ways to identify Alzheimer's early and work to protect our brains and preserve our memories. So this is a wordy title, but it's something that I spent a lot of time thinking about. Alzheimer's disease is not normal aging. It is an abnormal process occurring in the brain that causes cognitive impairment. And we use this term cognitive impairment because Alzheimer's may not only affect your memory, but other thinking abilities as well, such as language, planning, concentration. In clinic, these changes in thinking skills and other memory skills are confirmed on various thinking tests, like this one, the MoCA, or this one, the figure copy, or the class drawing. See, now you're all laughing because you go through much more vigorous testing than that. And that's because we need to have that vigorous testing. We need to have good data in order to do what we're doing. The ones I just showed you, those are just screening tests, but they're still valid. And after we rule out other causes of memory problems, such as medication side effects, depression, or the more common nowadays, sleep apnea, usually we identify someone as having mild cognitive impairment, or MCI, or dementia. Now, mild cognitive impairment and dementia are terms that we use to describe what we see in a person, but not actually what is happening in the brain itself. Now, Alzheimer's disease is one of the many causes of mild cognitive impairment. Alzheimer's disease is not the same thing as dementia. Dementia is a group of symptoms that we see in a person. These symptoms may lead to problems with different thinking abilities, like the ones I've listed here: memory, language, attention, insight, and this executive function. This is found in the front of the brain, and it's really what deals with multitasking, planning, organizing. The neuropsychologist here will tell you it's the ability to host a really good dinner party, all the skills that's needed. So dementia is this kind of umbrella term, as it can be caused by a variety of conditions, including Alzheimer's disease, but also vascular dementia, Lewy body dementia, frontotemporal, Parkinson's, or what we call mixed dementia, which is really just a combination of any of these, but primarily with Alzheimer's. Now, vascular dementia has traditionally been thought of as having memory problems after having a stroke. And this is still true, but we're starting to discuss this very real possibility that having enough chronic small blood vessel disease or narrowing of those blood vessels that are in our arteries could be enough to cause cognitive impairment without a stroke. Now, Lewy body dementia and Parkinson's disease dementia, these are also caused by abnormal proteins that can kill the cells in our brain. It's a different protein than the one that we'll talk about, and as a result, it has a different effect on how we're thinking and how we may present. For instance, people will have more Parkinson-like movements in these conditions. Now, frontotemporal is caused by another unique protein, and it affects the front and the side of the brain. And again, people will present differently. But these are all different causes of dementia. Now, Alzheimer's disease is the most common cause. It alone can account for up to 50% of cases. But when you consider Alzheimer's with any of those other ones, this mixed picture, it can cause up to 80% of cases. And not only can Alzheimer's disease cause dementia, but it can also cause mild cognitive impairment. So what do I mean by this? Well, in this picture, you'll see the horizontal axis, or the horizontal is age, and the vertical axis is your thinking ability. So to go from left to right, you're getting older, and to go from top down, your thinking is a little less effective. And notice how the black line is not completely horizontal. So we don't anticipate that everyone is thinking to the same ability as they get older. Unfortunately, this is what we notice. But you'll see that as we're getting older, some people are developing or accumulating these Alzheimer's disease brain changes. And these brain changes at some point can lead to mild cognitive impairment, a tipping point in which we notice these symptoms, and to dementia, at which point we notice some functional symptoms too. So we say that someone has mild cognitive impairment when they, or someone they know, notice a change in their thinking abilities, and that impairment or that change is strong enough that when we do that vigorous testing that you all have been through, we can find an impairment. At that point, though, a person can still compensate for those memory changes. They write lists, they use GPS. People develop many creative solutions in order to have good daily functioning. Now, we say someone has progressed to dementia when they can no longer compensate, whether it's the lists or the GPS, but they begin to have problems managing these daily affairs. Things like taking medications, getting lost, forgetting to pay bills. So in the RAP study in the field of Alzheimer's disease research in general, we really are working to try to spot these brain changes early in people before any of the memory tests or memory symptoms actually start. However, you can see from this picture that by the time a person actually has memory problems or thinking problems, these Alzheimer's disease related brain changes are already present. So that is why our RAP investigators and other researchers are using these cutting-edge technologies, this specialized MRI and PET scans, and unique brain chemistry patterns in spinal fluid to identify the earliest possible brain changes so that we can intervene and do something about it earlier. So what are these brain changes that I keep referring to in Alzheimer's disease? Well, if you read up on Alzheimer's disease, if you Google it, you may come across the terms plaques and tangles. Now, the term plaque is just a scientific, generic term for buildup of some substance. So you hear about plaques on your teeth and plaques in your arteries. When we talk about plaques related to Alzheimer's disease, we're referring to sticky deposits in the brain that are made of this toxic substance that we call amyloid. So these plaques are shown here in this figure, and then also on this slide. Now that's all happening outside of our brain cells, but inside of the nerve cells in the brain, we also see what's called tangles, or abnormal clumping of the substance called tau. So these plaques and these tangles, they cause inflammation and damage to the nerve cells in the brain. This damage leads to shrinkage of the brain over time, and that process we call neurodegeneration. Eventually, these brain changes lead to a decline in our thinking abilities, and eventually to our abilities in doing our daily tasks. So you will hear us refer to these plaques and tangles frequently in these presentations, as we're focusing our research in RAP on how can we spot these changes as early as possible. So a lot of research is now focusing on these three kinds of changes: A for amyloid, T for tau, and N for neurodegeneration, or nerve cell death. So our team is working together to see how can we identify these ATN changes in the brain and piece it together with what we see in the person, the most important part of this process. And by that I mean the cognitive test results, the health history, the family history, and the lifestyle history. And I end with that in particular because we are showing that there can be a meaningful impact in how we're living our lives. So we used to only be able to see plaques and tangles on autopsy. Now we can detect these early Alzheimer's-related brain changes decades before memory symptoms start. And it's by using this cutting-edge technology and measurement tools, primarily brain imaging and then the special tests that are done on the spinal fluid. And there are some in this room that know what that means to obtain spinal fluid. There's a snicker over there. All right. So MRI gives us a detailed picture of how the brain looks and whether there are brain shrinkage or signs of nerve cell loss. We can also see how good the blood supply to the brain is, which is an important thing that's pretty unique to UW and RAP. With functional MRIs, we can evaluate the connections between parts of the brain. And how I look at that is we're looking at the highways that connect to our cities. So PET scans can also show us different things depending on what chemical tracer is injected into our arm. If we have a glucose-related tracer, it can tell us how active our nerve cells are at using that glucose. It gives us a picture of any nerve cell injury. Now, if we use a tracer that sticks to amyloid, it can show us if we have any amyloid and how much we have. Similarly, if we use a tau tracer, it can show us if there's any tau accumulating in our brain cells. Again, the A, T, and N. So while there's not yet reliable blood tests, we can also safely collect spinal fluid from our low back area to measure precise levels of this amyloid and tau, as well as other substances that may tell us more about level of inflammation and nerve cell health overall. So in this picture, we have 3 separate MRI scans of the same person, but at 3 different time periods. And on the far left, that's time point 0. And in the middle, we have a 1.5 years later. And then the far right, we have 3 years later. So first, let's take a look at the top row. And notice how there's less gray color and more black as you go from left to right. It might not seem as prominent, but there is less of that. And what that indicates is neurodegeneration. So there's more brain cell death. Some of this happens as we get older, but we notice in Alzheimer's disease, it happens more. Now let's look at the bottom row. And what that really is, we've selected the memory part of the brain called the hippocampus. That peanut-shaped structure there is highlighted, and I'm wondering if you guys can appreciate. Oh, we can't have my mouse. Oops. This one right here. And so that's the hippocampus. And what we see is as we go from left to right, it also gets smaller. And 3 years later, it's pretty noticeable the difference in the size. So having this finding in someone who has cognitive impairment is very suggestive of Alzheimer's disease. Now this scan, as you can see there, so this is one person, 3 different scans. And on the far left, you have that MRI. So that's what we just looked at. And you'll see it's kind of hard, but there is a memory center that you can see there. And it doesn't look that shrunk. It doesn't look that small. At least from what I could see, if I saw this person in clinic, I would say maybe it's not as full, but frankly, it's not strong enough for me to think that it's actually atrophied. But then you take a look at the middle picture. And what that is showing is the presence of amyloid. And you see that it is present throughout the brain. And then you take a look at the one on the far right, and that one's showing tau. So the tau signal is lighting up, and it's also showing the presence of tau tangles. So these pictures were taken in a RAP participant who did not have any memory loss symptoms. So does that mean this person has dementia due to Alzheimer's? The answer is no. They don't have any symptoms. They don't meet our clinical diagnosis of dementia. However, they are at increased risk for dementia in the future. But we don't know if or when they will develop it. So that's one thing we're trying to learn so that we can provide patients and their families more information and more accurate information. Now, spinal fluid tests can detect these very early changes in amyloid, tau, and neurodegeneration. Think of it that imaging tells us where these changes are occurring, and spinal fluid gives us a more precise detail about how much changes have actually occurred. And in fact, they could potentially tell us changes that are occurring 1 to 2 years before imaging, although imaging is becoming more and more advanced, so maybe they will catch up. But if you think about it together, brain imaging and spinal fluid are very powerful research tools. And it's these tools that we're using in our Alzheimer's prevention studies. And this is important because our current FDA-approved treatments for Alzheimer's really treat symptoms and not that underlying disease. So the medications currently used really help dying nerve cells communicate with each other for longer, but they don't address the amyloid and tau that we've been talking about. And we use them because they can help with symptoms. There can be a meaningful benefit, but it does not address the underlying process, and so it cannot stop the process of decline. And the two main groups are cholinesterase inhibitors, and then I'm not even going to try that word again, but I'll just say NMDA blockers. And cholinesterase inhibitors, with Aricept being the most common, they act in a way that allows the chemical signal in the brain to be around longer. So having that signal around longer increases the chance that cells are going to communicate to each other. They're going to tell each other to remember X or try to remember Y. But they don't create the signal, and eventually it's natural for that signal to be broken down. We can't have those signals all the time. Now, Namenda is our only NMDA blocker, and it tempers the movement of calcium that goes in and out of our brain cells. And this is important because too much calcium can create what we call excitotoxicity. If there's too much excitement, the cells get sick and they stop working. And I think of this as when we drink too much coffee or we drink a cold slushy too fast. We feel the effects of too much. And so that's what's happening in our brain cells. Too much of something can cause harm. So cholinesterase inhibitors have been around for a long time. Donepezil was approved in 1996, and memantine is the newest drug, and it was approved in 2003. So we haven't had a new drug in 15 years. And I think we can all agree that is simply unacceptable. So in this figure, I just wanted you to see the traditional figure of what it looks like to have this progression. And right here is where we would do symptomatic treatment. And we should be able to do it much sooner. We should be able to address this earlier in the course, which is what we're trying to do with our clinical trials. So clinical trials in the recent years have been studying ways to reduce amyloid and tau and those changes. But the first step is to identify people who've had these changes, but are still having normal thinking abilities. So that is why brain imaging and spinal fluid biomarkers are so important. If we give a person a therapy that reduces amyloid buildup, but they don't actually have amyloid in their brain, the therapy isn't going to help them, and it's going to be deemed to be an unsuccessful study, when in fact, we really may not have been studying the right person at all, and we had bad data to begin with. So there are studies that are doing it the proper way. An example is this well thought out study called A4. And this is a worldwide study which UW is a part of, in which older adults with normal cognition are screened with amyloid PET scans, and only those that have an elevated amount are eligible for enrollment. And once you're in the study, you receive either an anti-amyloid treatment or the placebo. Now they finished their recruitment and the results should be out in the next several years, but you can see they were very successful in getting almost 1,200 people enrolled, all over the age of 65 to 85. Now they do have to come into our hospital every 4 weeks to receive IV treatment, but this idea that we really need to treat the right people is very prominent in them, and they're grateful to be a part of the study. We're looking for their cognitive change to see if what's happening over time, and we need them to do this. Now, Alzheimer's prevention trials are also using this imaging and the spinal fluid tests because we need to identify who has increased amounts of amyloid, tau, and neurodegeneration. And for those that are already in studies, well, are we seeing a difference in these biomarkers? Are we seeing changes in their amyloid tau? So RAP and other studies are really paving the way toward more personalized medicine, which I think is what we all want. And they're doing that by identifying the best markers to monitor our brain health in cognitively unimpaired people. Now, other ongoing studies are looking at drugs that may reduce amyloid production or improve its clearance from the brain, therapies that may stabilize or reduce tau, medications that could reduce inflammation and or protect our nerve cells altogether. And then lastly, the one that I think is really important is non-medical strategies that could reduce amyloid and protect nerve cells through improvements in lifestyle factors like sleep, exercise, and diet. And the side effects to these are high quality of life and improved mood and better function. A little bit different than the medication ones. So RAP is really a part of a worldwide initiative to better understand what is Alzheimer's, what can we do as early as possible. And we're in really good company with other centers, other institutions in the United States, in Asia, in Europe, and in Australia. And while I don't have quantitative evidence to prove it, I do believe we are the best study out there. I don't think anyone here is going to refute me. And really that is because of you and all of the work that all of you are willing to do and willing to give to us. So thank you for everything that you do for us. And please consider doing more.
Well, it's lovely to see you all tonight. My name is Lindsay Clark, and I'm a neuropsychologist who works on the RAP study. And tonight I'm going to be talking about some findings from RAP, so getting into a little of the science that we've been doing lately. But before I jump into that, I want to just remind everyone what are the overarching goals of the RAP project. We're trying to identify Alzheimer's disease in its preclinical phase before cognitive symptoms appear, and to identify biological, health, and lifestyle factors that may increase or decrease risk for developing this disease. So to achieve these goals, it takes really dedicated people like yourselves to provide a lot of data, to come in and do the cognitive testing, to do the blood draws and physical exams, to fill out all kinds of questionnaires about what you eat, when you sleep, who you hang out with, all kinds of things, and to come in and donate some cerebral spinal fluid or let us look at your brains, and also takes a large research community to put all of this information together in a way that makes sense and helps us to answer these questions and achieve these aims. Because of this, we've been able to publish over 110 manuscripts using RAP data. And so in this section, we're going to try to highlight some of these findings from a recent review of the RAP study that Sterling Johnson published. Some of our early findings looking at biomarkers and how they relate to cognitive function. Some of the changes or associations we see with lifestyle factors such as physical activity, cognitive activity, diet, sleep, the microbiome, stress, how your environment or neighborhood may affect your thinking, and also how we're trying to be a little more sensitive in detecting with in-person cognitive changes. So before I jump into that, as Sterling mentioned, there's over 1,500 participants in the RAP study currently, and over 1,200 people are actively enrolled, meaning coming in every 2 years for the evaluations. We, on average, have individuals who are 64 years of age and have been in the study for 9 years. And about 40% of people are at genetic risk for Alzheimer's disease, meaning they carry at least one E4 allele on the ApoE gene, and 73% have a family history of Alzheimer's. We are also working to continue to increase the diversity of our sample by encouraging diverse participants so that our study results are generalizable to our whole population. So, as you may remember, the study was started really with the hypothesis that having a parent with Alzheimer's disease increases one's risk for developing the disease themselves. And in some of our recent studies, our recent analyses, we've been looking to see if people who have a parent with the disease show cognitive changes at faster rates than people who don't have a parent with the disease. And so far, what we have seen is that people who have a parent with the disease, who are shown in this graph by the blue and brown lines, do not show faster rates of change on our neuropsychological measures than those who don't have a parent with the disease. Similarly, people who are at higher genetic risk for Alzheimer's disease, having the ApoE4 allele, also do not show faster rates of cognitive change on our tests over time. So what this suggests to us is that we need to find more specific markers of disease risk, such as what Dr. Chin was talking about, looking at specific markers of the amyloid or tau proteins in the brain. We've also observed that more than 80% of our participants are cognitively healthy, so remain cognitively stable over the course of the study. And only less than 4% have progressed to clinical levels of impairment, meaning developing some of those clinically significant symptoms such as MCI or mild cognitive impairment or dementia. We have about 15 to 20% of the sample or of the participants in the study who have exhibited maybe some subtle cognitive decline, which we're still trying to understand if this is clinically meaningful. We notice that this category does fluctuate with time. So individuals may come in 1 year and look like they're impaired relative to their peers, but then come in the next evaluation and bounce back to normal. So this is still not quite explained and is something we're still exploring. And one of the ways we're trying to explore this further is to be more sensitive about identifying within-person changes. And this is using the idea of extending growth curves that we often think about in pediatrics to help us identify preclinical cognitive changes. So for example, on the left of the slide here, these are growth charts, like height and weight charts that we see for ages 2 to 20. And you can see a red line here and a yellow line here, which are individual people superimposed onto these population-specific centiles. And on the right side here, we have kind of similar growth curves for our memory tasks. And each of these lines, these dotted lines, represents a percentile of the population, a demographically specific percentile. So typically what we'll do is we'll notice that if people are performing below the 7th or 2nd percentile relative to similar peers, similar in age, gender, educational background, for example, we might note that that person may be performing weaker than we would expect relative to their peers. However, one thing that this approach does not take into account is where people start on those tests and whether there may be a more significant change in where they started. So I'm going to walk through an example to explain this a little bit more. This is an example of one participant in the RAP study's performance over 4 visits. And we can see at visit 1 that this person started at about the 84th percentile compared to their peers, so very above average. And by visit 4, they were performing at the 25th percentile. So compared to their peers, they're still performing within the normal range, greater than 25th percentile. However, when we take into account where the person started and include that into our algorithm and compare them to their peers and also where their peers started on this test, we actually observe that this person is in the 6th percentile. So that suggests a potentially worrisome change for this person from their relative baseline. We also notice on this graph that we have marked here that this person recently had an amyloid PET scan in part of the biomarker studies that was significant for elevated amyloid. And so we can see not only has this person shown a significant change from where they started, but they also have some of this elevated amyloid protein in their brain. This is a second example of a person who doesn't show a significant change from where they started to where they are at their most recent visits. They've bounced a little bit, but when we use kind of this algorithm that takes into account where they start, they're still performing well within normal ranges. One thing that's interesting with this case is that this person also recently showed elevated levels of amyloid protein on a PET scan. And so this is actually an interesting case as well because it helps us to start learning about aspects of cognitive resilience. How is this person able to continue to perform well above the normal expectations in the face of having some elevated levels of amyloid in their brain? So this approach represents a new technique for our field, and it's generally simple to interpret and may have some clinical usefulness after we continue to provide some more validation, and we hope to expand it to some of our other cognitive measures that we're using in the study. Additionally, we're going to be trying to integrate some of these measures or more sensitive measures of cognitive change with biomarkers of the underlying AD pathology. So I mentioned at the beginning that at this point it does not seem that family history of Alzheimer's disease or the presence of the ApoE4 allele is associated with greater rates of cognitive change at this stage. However, we have found in some of our early studies that elevated levels of biomarkers, particularly amyloid, does seem to be associated with some significant cognitive changes. So this is, I'm going to talk about a study that we recently published where we analyzed the cerebrospinal fluid data that people donated by doing a lumbar puncture. And we analyzed that data in a way where we were able to characterize individuals as having elevated levels of amyloid in the brain, elevated levels of tau, or elevated levels of both amyloid and tau. And about 70% of the sample were biomarker negative, meaning they didn't have elevated levels of either of these proteins. And we found that the number of participants with and without amyloid didn't differ in terms of their family history of the disease. However, one of the important findings that we observed with this study is that we noticed that people who had elevated levels of amyloid, as shown with this orange line here, or elevated levels of both amyloid and tau, as shown by the green line, seem to show more significantly faster rates of change on our cognitive measures over time than the individuals who didn't have either protein elevated or who only had the tau protein elevated, shown here by the blue line. We also looked at a separate sample who had the similar data, but looking at the amyloid PET scans, and we found similar pattern of results, whereas those with elevated amyloid on their PET scan also seemed to show greater rates of change on some of our neuropsychological measures over time. So we're continuing to look at these data, and in some of our further analyses, we've begun to observe that these declines may not be across the whole group of people who have elevated amyloid, but may be more apparent in individuals who have both elevated amyloid and other risk factors such as hypertension or obesity. So for example, in this analysis, we found that individuals who had both elevated levels of amyloid and hypertension, shown here in the green line, declined faster on some of the cognitive measures compared to those who had elevated levels of amyloid but no hypertension, or those who didn't have elevated levels of amyloid. So this is a really important follow-up study because it suggests that there may be other factors that we can potentially intervene on to slow the effect that amyloid may have on cognitive change over time. And so to talk about more of those health behaviors and how they relate to cognition and to some of the biomarkers that we're looking at, I'd like to introduce Dr. Okonkwo, one of the other researchers on the RAPT study.
All right, so the first study I want to talk with you about is a project looking at how physical activity might be a modifier for risk for Alzheimer's. So in this study, we put you all here through some questionnaires that helped us assess how physically active you all have been. And we also had you undergo some PET scanning that looks at the amount of amyloid in the brain and also how your brain uses glucose, which is the primary fuel for the neurons in the brain. And what we had observed is that when you look over here, this is age going from 45 to 75, and then this is amyloid going from being low to being high. When you look across everyone on this plot, what you see is that as you increase in age, there is an overall increase in the amount of amyloid that we detect in the brain. However, the folks in this study were classified into two different groups. Those whom we deemed to be physically active, that is that they met the American Heart Association guidelines for about a half hour of moderate intensity activity 5 times or so every week, compared to those who did not meet that guidelines. What we see is that the increase in amyloid is more pronounced in those who were physically inactive, so those who did not meet those guidelines. Those who met the guidelines seem to kind of stay stable over time, and so did not show increased amyloid with aging. The reverse was observed with glucose uptake in the brain, and in this case, higher amounts of glucose uptake in the brain is good. So what we see again is that as people aged, those who were physically inactive tended to show less glucose uptake in the brain compared to those who were physically active. And in case you're wondering what do we mean by moderate intensity physical activity, it's going to be different for everyone just because we're in different physical conditions. But one way to think about it is that intensity or type of physical activity that when you are engaged in it, you can hold a conversation with someone but probably cannot belt out a song. So that is how you know that your heart rate is in a moderate intensity range. And then the next study, I'm still looking at physical activity. This time around, while we looked and used a questionnaire in the prior study, in this one we looked at a direct measure of how physically fit an individual is. And some of you have been in this study where we put you on a treadmill and have you run until pretty much you pass out. Is this study here? So here we split individuals into those who were less fit and those who are more fit. And then also we quantified each individual's level of genetic risk for Alzheimer's. And those who are in the red bar are those who we deem to be at high genetic risk for Alzheimer's. And what we see here is that if you look among those who are less physically fit or aerobically fit, you see what you would expect, that when you are at higher genetic risk for Alzheimer's, you have an increase in this marker that we get from CSF, a marker for risk for Alzheimer's. So it's higher among those who are at increased genetic risk for Alzheimer's. However, if you look among those who are more physically fit, you do not see that same effect. So what this suggests is that aerobic fitness might somehow attenuate one's otherwise genetic risk for Alzheimer's. So moving on from this study then, our colleague Dr. Bendlin is doing a study called the Microbiome and Alzheimer's Risk Study, MARS. And a bunch of you here are also in this study. And so this study, the goal here is to understand how the bacteria in our gut might have a role to play with risk for Alzheimer's. And as you probably know, our bodies are pretty much, petri and dishes. So we carry a bunch of germs and bacteria in us. And so the goal here then is to try to understand whether there is an association between both the different types of bacteria that we harbor and also the amount of each type. Do those have anything to do with our risk for Alzheimer's? And to accomplish this, we sent home with some of you kits to enable us collect stool samples that we then assay and quantify for these types of microbes in the gut. And initial findings from this study suggest that when you compare people who have dementia of the Alzheimer's type to age-matched, cognitively healthy adults, that those who have dementia from Alzheimer's, there is a reduction in the diversity of gut bacteria that they carry. So they do not have as diverse of gut bacteria. Now, when we moved on then and looked at people like you who are cognitively healthy and mostly in midlife, we looked at, is there an association between the abundance of specific types of gut bacteria and a CSF marker for Alzheimer's? What we see here is that people who have more of a specific type of gut bacteria show an increase in the amount of amyloid in the brain. Now, this is all still findings that we're still looking at to more fully understand what types of bacteria these are and what do they tell us about how the brain functions. In addition, the findings I showed you are probably, as what we call correlational studies. So it's telling us that two things go together. It's not telling us, though, if one thing is the cause of the other thing. And so we need more studies that can begin to drill down into mechanisms that tell us how specific types of bacteria might be a cause for Alzheimer's. And so for this study, our colleague, again, Dr. Bendlin, is partnering with some of our other colleagues to look at mice who were bred without any germs, and then they are going to be exposed to specific types of bacteria to see what their risk for Alzheimer's is. In addition, the study I described in folks like you here is continuing and is ongoing, and so the hope then is to follow you all over time to see if the types or diversity of bacteria in one's gut at a point in time can tell us something about risk for cognitive decline and possibly progression to MCI or dementia over time. Then for this next study, here we looked at the questionnaires, which are numerous that you all complete, that ask questions like, what types of games you play, physical activity, your diet, your sleep. And in this study here, we are trying to look at all of those measurements or factors together to see, how do they jointly contribute to maintenance or decline in cognition and then progression to dementia over time. For this particular slide here, though, what we're looking at here is engagement in cognitively stimulating activities like playing cards, puzzles, reading, and so forth. And so here we divided the sample into three groups. Those in blue are those who engaged in cognitively stimulating activity very frequently, and those in red are those who did so least frequently. And what you can see here is that the decline in cognition over time is more pronounced in those who are not as mentally active, whereas those who are most mentally active actually seem to have something of, a little bit of a practice effect. So they benefit from seeing this cognitive test over and over again. And so it looks like their scores are getting a little bit better, but it's just what we call practice effect. And that's a good thing. It's a good thing that you're able to record that I've seen this test before and I kind of know what the answers are. That's a good sign, actually. All right, we're going to switch gears again because we're trying to give you a sampling of different aspects of our findings. So I will switch gears once more and now talk about sleep. I think everyone here would agree that being able to get a good sleep is a good thing, right? And we are less cranky in the morning if we get a good night's sleep. But it turns out that sleep is not only good for your mood, but it's also good for your brain and cognition and has a role to play in risk for Alzheimer's. So this is work from Dr. Sprecher, who was a grad student in Dr. Bendlin's lab, using questionnaire and data that you all here provided that looked at how adequate of a night's sleep you get, what types of sleep problems do you report, how well rested you feel in the morning when you wake up from sleep. And then to relate that with those cerebrospinal fluid biomarkers for Alzheimer's. And these findings are remarkable because here she shows that when you look at either the adequacy of one's sleep or how sleepy one is during the day or the types and the number of sleep problems one endorses, that each of those is associated with these CSF markers for Alzheimer's. So feeling more rested after sleeping is associated with having reductions or lower levels of these markers for Alzheimer's, whereas those who report having more sleep problems show higher levels of these CSF markers for Alzheimer's. So you probably would agree that just as having good sleep is something that we should all aspire towards, Having stress in one's life is something that we all would like to avoid for all kinds of reasons, right? And here, our colleague, Dr. Zulsdorf, has done some work to show that not only our stressful experiences something that affect our mood and how we feel, but they also have the potential to affect how we think or how we perform on thinking tests. And so for this study here, she looked at a particular measurement of thinking skills called speed and flexibility. So how fast we can process information. And what she shows in this study is that when you quantify the number of lifetime stressful experiences an individual has had, and you relate those to how quickly they are able to take in and process information, that those who report more number of lifetime stressful experiences show poorer performance on this measure of of speed and flexibility. So it indicates that stress might be one of those modifiable lifestyle factors that if we take care of, it might play a role in reducing our risk for cognitive change. And then similarly too, our colleague Dr. Amy Kind has also looked at neighborhoods where individuals live and how that might have a role to play in cognition. And just as with the prior slide, she's also finding that individuals who live in disadvantaged type neighborhoods show poor performance on measures of cognition. Again, these are all findings that are still kind of new, and we're yet to fully understand what they all mean. We have on this slide now just a listing of some of the other types of modifiable lifestyle factors that each of us could look at and see which ones are we able to make changes to in our life, because all of these have been shown to have some type of role to play in our risk for Alzheimer's. I'll wrap up this section by just again thanking all of you here for your time and your effort and your commitment, because none of this data that we've shown tonight will be possible without your commitment and your showing up time and time again to give us more and more and yet more. So thank you. Thank you very much.
Well, you've seen a lot of data now, and this is truly remarkable stuff. We wouldn't have thought necessarily 5 years ago that this family history effect that Dr. Clark mentioned wasn't going to have an effect on cognition. I think we all thought it may have, because early on we were seeing some brain changes with that. But it turns out that it's not influencing cognition, at least not yet. Neither is ApoE. We know that ApoE is a important genetic factor, and I don't think those of us who might have that gene are off the hook necessarily. It's just that the sample, you all are still relatively young compared to the age of normal onset of Alzheimer's disease. So we need to keep following that. But I think what we also learned is that the biomarkers, amyloid and tau, are in fact more specific to this disease, and they are showing effects. And then importantly, we learned from Dr. Okonkwo that there are things we could be doing about it in our lifestyle and health that may in fact alter the trajectory of Alzheimer's disease. So this is really incredible. I'm going to just try to sum up a little bit here and give you an update on the status of the project in general. I'll go over the NIH grant, the registry part of RAP. RAP is a registry, and much of what we saw tonight was because you've participated in linked studies to the main study. And let me just circle back to the goals of the study, identifying this disease as early as we can and identifying those features that might confer risk or resilience. And this study is a longitudinal cohort in and of itself, and it's a valuable cohort that is being looked at all over the country, and in fact by investigators all over the world. And again, our success is because you all are willing to participate in the registry part of it, and when we have a study come along that's linked to our main study, we give you a call or send you a letter, and you generously participate. in some of those linked studies. So otherwise we wouldn't have found what we found about exercise or the microbiome work or the stress work or the neighborhood work. Those are all really incredible linked studies to us, including the amyloid imaging that we've done so far has all been in a linked study because we haven't had the funding in the main RAP program to do those kinds of studies centrally within the main part of the study. So that is going to be changing. So let me just refresh this slide, refresh your memory on this. We know that we want to identify this disease as early as we can before these cognitive changes in red happen, and we can do that through these specialized biomarker tests that we have the capacity to do right here in Madison. And it's only with this new understanding of the disease that we didn't have when we started this project in 2001. We didn't have tests for amyloid back then, but it's only with that kind of precision in understanding the pathology of the disease that we can finally address these questions like, does sleep or physical activity prevent or slow AD? And you noticed in that slide that Dr. Okonkwo showed at the end, what can you do, what can you do to raise your resilience and lower your risk? We didn't have any numbers on any of those slides. I'd like to be able to say to you that if you exercise for 30 minutes a day, 5 days a week, you can lower your risk of Alzheimer's by X amount and put a number on that. I think we can probably do that by the end of this study because we'll have had such detailed information on you, and we'd like even more of you to participate in biomarker studies, and that will link these lifestyle features to the pathology so that we can really get a better understanding of all of this. So far we have some really important correlations. They don't confer causation, as you know, but they are something that we're all paying attention to. And as these correlational association studies show up repeatedly over and over again throughout our studies, we're really paying attention, and we see that there's some meaningful information here, thanks to what you're providing. To answer RAP's fundamental questions about lifestyle and biology being the genetics and your other health features, to answer these key questions, we need to measure the AD pathology itself, and we can do this now. We need to know who has evidence of brain evidence of Alzheimer's, at what age does this start, and how fast is it accumulating. And we need to know if any of those health behaviors that we've talked about influence any of this. Does somebody who has a healthy diet or exercised, does their amyloid happen later in life, or is it possible to that one could stave off this disease with some kind of lifestyle intervention? Those are things that we'll be able to study as we go forward. Well, as we've talked about before with Dr. Chin's talk, the way we've diagnosed AD in the past has been under a microscope, and now we can do this with these specialized scans and with spinal fluid tests, which we're doing. And as I mentioned, the studies so far have been in these linked studies in the registry component of RAP, in the studies like PREDICT and studies like FAB and LEAD that have had CSF in them or amyloid and tau scans in them. But now we have new funding from the NIH that'll go till 2023, and that's important news. So thank you for that. That's worth a clap. It's really thank you. It's a relief for all of us investigators to have the funding now. But importantly, what we proposed in this, and hopefully you're willing to help us with this, is that we'd like to expand our CSF, our spinal fluid program, and our amyloid and tau imaging program to the main study itself. And so that's what we'd like to do. So the new study elements will be that in addition to your main study visit, we'd also like to schedule you on a separate day for consideration of CSF with an MRI, or those PET scans and an MRI, or all three of those things together. And These things are always optional. As with everything in the RAP study, you don't have to do any of it, but we appreciate you would think about raising up your level of intensity in your participation in the study to some of these biomarker types of scans or assessments. It's really with these kinds of things that we can understand the biology of this disease in more detail. So we'll be starting this this summer in the main study. We'll be, as we schedule you for your visit, we'll be asking if you would consider the spinal fluid procedure or the PET scans or both. And you can say yes or no to those things, and that's totally fine. I also wanted to just highlight here some of the RAP procedures that you go through in your main study visit. We know you do so much. You go through the cognitive testing, you know what that's like. We've added computer testing to that to add insult to injury. We've asked you to do more testing on the computer, and that's because you know those other tests so well that we need to give you something else. And in fact, that's true. Those computer tests are less susceptible to practice effects that we heard about. They are more culturally fair, and they are also independent of the diagnosis that we would make using our regular cognitive tests. So those are really valuable procedures to us. The medical and physical exams that we do are really important to help us understand other causes of cognitive impairment. Alzheimer's disease is only one cause of cognitive impairment with age. It's the main one, but it's only one. And you continue to do the laboratory tests with the blood that we collect from you. The genetics is really important. Dr. Engelman is here. She's our geneticist, and she'll help answer questions later on. Of course, those questionnaires are fundamental to understanding lifestyle as you report out to us what you've been doing and what your health behaviors are, and study partner questionnaires as well. And these things in red are some of the new things that we are going to do that I just mentioned to you. There's also a new partnership with the National Cell Repository for Alzheimer's disease, and this is a national bank, basically, that we would like to contribute some of your blood to and some of your spinal fluid to, and we'll explain that to you when you come in, and you'll have to sign a special consent for that, and we would explain all that and make sure you're okay with that. But that's so that other researchers around the country can get at your samples, which are very precious and valuable samples. This study is one of the major studies in the country on preclinical disease, and so your data and your samples are really valuable to other researchers throughout the country who could probably do things for some of these things in a way that we don't have the expertise here. So just let me dwell a minute on the registry part of RAP. Here's a list of some of our linked studies, and many of these studies are in the back of the room here, including our PET imaging, which we're doing right now with the PREDICT study, and also there's a whole list of things here. The microbiome that we talked about is there, the exercise studies, the GROW study, which is a study of families. If you have a brother or sister or parent who we can get genetic information from, we would love to talk to you about that, and that's, again, in the back of the room here. And there's also some partnership studies here. We're partnering with other adult children's studies. There's one at WashU in St. Louis. There's one in Baltimore. We're partnering with these studies and sharing data and trying to come up with answers as fast as we can by pooling the knowledge that we have and the data points that we have. This slide just goes over some of these different facets of our study and our program of studies, really. Lifestyle and physical, starting on the lower left. Health equity is important to us because we want to make sure our results are generalizable to all groups. Medical comorbidities and vascular disease, imaging markers, fluid biomarkers, genetics, metabolomics, which is an assessment of all the molecules in your blood related to your energy metabolism and other metabolic processes. And we bring all this together with computer science. This data is immense, as you can imagine, and our computer scientist friends on campus are highly involved. We're part of a major center in the computer science world where we're utilizing this data with computer science algorithms, machine learning, and deep learning, the same thing that Google and Uber are using to help figure out how to make a car drive itself. We're using that kind of technology to help us understand very early and small or weak signals in the data that we have. It's really important and very interesting to work on. So things on the horizon as we look ahead, now that we want to do more of our biomarker studies, we would like to be exploring how we can get these results back to you. And we want to do that in a very thoughtful way. We're not ready to tell you about your results. In fact, the results aren't really interpretable yet because we have to build the house before we can live in the house, and we have to figure out what is the normal amount of amyloid, and is it predictive? And once we can put some precision on that, we will get that information to you. We're not trying to hold on to any of this, but we want to make sure it's clinically relevant and meaningful, and we're not worrying you needlessly over signals that we might be excited about, but in reality may not mean much. And so that sometimes takes time. So we appreciate your patience with us. And there's a couple other things. I mentioned the National Cell Repository, or NECRAD. There's an International Alzheimer's Prevention Initiative that we're a part of. This is a group that is doing drug trials in people at genetic risk and people with various other risk factors. And we are not doing the intervention part of that study, but we're partnering with them and understanding rates of change so that they can understand, they can take what we're learning about rates of change in people who are amyloid positive and negative and understand what it might look like in a clinical trial if that clinical trial actually worked. How would it modify that rate of change? And of course, we're very interested in a blood-based marker for this disease so that we don't have to put you through or put the next generation of participants through all of these spinal taps and lengthy PET scans that we have to do to get at this disease right now. So we're able to do this kind of work because we have incredible scientific and supportive infrastructure for RAP. We're supported by the investigators who do these studies, and many of them are here tonight. We're supported by the philanthropy of the school. We have financial support through the Wisconsin Alzheimer's Institute and through the NIH. We're supported scientifically through the Alzheimer's Research Center, which provides the facilities to do the spinal fluid collection, the imaging, and many of the other informatics and data things that we do. And we're supported by the School of Medicine itself, who's been instrumental in keeping RAP going over the last 5 years. This is a busy slide, but there's two columns here. One is our Wisconsin ADRC, or our Alzheimer's Research Center, and the right column is the registry, that's the RAP study. And I put these side by side because RAP exists synergistically and coexists with our center. Our center has patients with dementia in them, and if you have a loved one with dementia who wants to be in research, you can refer them to our Alzheimer's Research Center. But I mention it here because the data from the center is going to help us interpret your data, because we have the cases with dementia and the controls, and that will be used to help us develop thresholds and guidelines for what is normal and what is not normal on some of these biomarkers that we're collecting, and then we'll apply that to the information we're collecting from you in the RAP study. And I'll just close with an initiative that the med school has been putting together called the Initiative to End Alzheimer's Disease. And the SMPH, or the School of Medicine and Public Health, has declared that Alzheimer's disease and cancer are its two top priorities. And in this initiative to end Alzheimer's, we're combining the two major hubs of activity on campus, the Alzheimer's Institute and the Alzheimer's Research Center, we're combining these under one umbrella, this initiative to end Alzheimer's, to bring and to coalesce our programs from bench to bedside, to bringing that knowledge to the community and to you and to your primary care physicians to better treat this disease. So the focus of the initiative is on finding people at risk. That's where you come to play. Discovering ways to treat and prevent this disease, providing care, clinical care to people in the state, reducing health disparities, and developing and disseminating innovative community programs with this. And you have this handout here in your slides, and also on that blue sheet of paper is the website here for the Initiative to End Alzheimer's Disease. If you're interested in learning more, it's at memoriesmatteruw.org, and that's through the Wisconsin Alumni Foundation. So I'll just conclude by saying that RAP is thriving and producing new knowledge because of you. It's happening at a fast pace, and we're scientifically in the pocket. We're scientifically centered on answering the fundamental questions about risk and resilience for Alzheimer's disease. Biomarker collection is something that we didn't think about in 2001, but now it's really where the field is, it's where the science is, and it's a priority for the study now. And we'd like you to consider participating in those biomarker studies. And finally, we're sharing data and the results of this study throughout the world, and together with other studies and other talented scientists throughout the world, we think we'll be able to find answers faster to this disease and finally prevent this disease, as our name says in the Wisconsin Registry for Alzheimer's Prevention. So let me stop there. We are, well, let me just thank you all for all that you do, and this work wouldn't be possible without what you do. So thank you.